So 3 out of 4 classes got a little bit further than this, so it may be a review for 3 out of 4, but I want to start my last period. Just got to the end of interphase. The things that you see on this picture for interphase, the form of chromosomes, which are chromatin, the nuclear membrane is present, and then these centrioles, which will aid in the process of division. You're going to see more today, or this video, what their role will be. They have replicated. Remember, this is also replicated. The DNA, it's not in this form yet, but you went from one chromosome or one DNA strand into an exact copy of it. So the next thing we're going to do is go into the second round of the cell cycle, which is the mitosis. So you should be at this point familiar, 3 out of 4 classes, prophase, metaphase, anaphase, and either telophase or telophase, whatever you prefer. We're going to go into, this is now the active time in which your nucleus is going to divide. The nucleus divides technically before the actual cell divides. There are 2 different stages. The last stage, which is stage 3, is when the cell decides to go from 1 cell to 2. This is when we go from one nucleus, or one content, whatever's in the nucleus, into 2. So we're going to first focus on prophase, and 3 out of 4 classes already got there. So this should be review for you. Where we left off, here's your cell membrane. During prophase, you can say the nuclear membrane disappears. I don't think I spelled that right. But that dotted line represents the disappearing nuclear, that phospholipid bilayer that probably is the nuclear membrane as well, disintegrates. It goes away for this process. The centrioles, which were produced and replicated during interphase, will begin to migrate. I'm not going to put them fully because they won't get there completely, but they're going to start at the beginning of prophase and continue to migrate. So the centrioles migrate to what is called the poles. The poles are just the ends or opposite sides of the cell. They could be here and here, they could be here and here. It doesn't matter where I don't think they distinguish what sides, but they're on opposite sides. The last thing that happens is going to be that chromatin, which looked like that spaghetti in the last slide. That's going to start to organize itself, and it will organize itself into this replicated state. Recall in interphase S phase, you went from one to another via the synthesis. It copied itself. So now it looks like the letter, you can see maybe in the shadow, it looks like the letter X, and that's how they generally draw these chromosomes. So I'm going to draw them, keep them consistent with all my classes. So you're going to see, I'm going to draw 4 of these X's. Each of these X's, this X right here represents one replicated chromosome made of 2 sister chromatids. So in this disappearing nucleus, I have 4 replicated chromosomes. I've got 8 sister chromatids. 1, 2, 3, 4, 5, 6, 7, 8. You don't generally refer to a number within a nucleus in number of sister chromatids. You'll recognize the number of chromosomes you have. So in this case, they're replicated, but we still have 4 replicated chromosomes. And you have 4 centromeres that are connecting all of the sister chromatids together. So this is the last thing. You can say the chromatin, the word that they will often use is the word condenses. Remember from the last video, what helps DNA condense? It's that vocab word, histones. Refer to the first video, or from yesterday, to remember what these are. Alright, I'm not going to go into detail. Chromatin will condense, and with the help of the histones. That was something that no classes talked about, but as a review, you should be looking or thinking about that. So then what we'll do is we're going to move on to the next one. So all of you now are on the same boat. The next phase is something called metaphase. Meta means middle, so there is something that's going to deal with the middle of the cell. So here's what we got. There's our cell membrane. Our centrioles have completely moved to the opposite side or the opposite poles. This is one thing that happens at metaphase now is the movement of the replicated chromosomes. The replicated chromosomes will migrate, they're like little birds, to the equator. I wish we all were there right now, feeling that nice warm weather. But they call the middle of the cell the equator, just like they call these the poles. They'll call this the equator. So I'm going to take my replicated chromosomes and I'm going to arrange them at the equator. Keeping it consistent, I have 4 replicated chromosomes here, so I'm going to take those 4 and line them up. There's 1, 2, 3, 4. I did draw them different sizes. You see 2 of them look similar, 2 of them look similar. This concept will be talked about eventually, but it's not necessary at this moment to really go into that detail. The question is, how did they get there? They got there via these centrioles. Centrioles are responsible for cell division in the fact that they make a substance called spindle fibers. So the spindle fibers will attach. So you can draw spindle fibers coming out of the centrioles. They're like little spidey webs. So if you're like a centriole, it's like, I'm going to go attach myself to a chromosome. So it goes tsh, 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 tsh. It's attaching itself to those replicated chromosomes. But this one is also doing the same exact thing. It's going to be like a little tug of war in the end. So this spindle fiber is brought to, there we go. Technically, these spindle fibers are attached, or they attach themselves at the end of prophase, because they are the ones that move them to the equator. They didn't just line them by themselves. They were helped after they were attached to the spindle fibers brought them to the equator. And that's all that happens in metaphase, is that these chromosomes get aligned at the equator as such. If this were a human cell, there would be 46 of these across the equator. I don't have that much room. I'm keeping consistent with 4 replicated chromosomes. That's all you need to know. Metaphase is that simple. The next thing. Anaphase. Now what will happen in anaphase, my centrioles are still at the poles, but what's going to happen is the spindle fibers are going to begin to retract. So imagine the chromosomes are lined up the equator, and I take my spindle fibers and I start yanking them towards the centriole. And the other centriole on the other side of the room that you can't see is doing the same exact thing. What will happen to the replicated chromosome? Imagine I've got an attachment on one side, and the other centriole has an attachment on the other side, and we start to pull. It's going to snap. You can see that. That's what would happen. So my replicated chromosome, which was connected via the centromere, is split into. My sister chromatids are no longer together. So we're going to draw that. And the way that they usually draw it is, so there's like 1/2 So why do they look like little V's? Imagine if I yanked this chromosome, you could see it would kind of bend, and that's the whole idea. It's going to bend like that, and so that's one sister chromatid being pulled, the other sister chromatid being pulled the other direction. So if you want to put this in words, if there's the diagram, you can look at it, get a sense of what it is. There's different ways you can say it. You can say the replicated chromosome is split. That's one way to put it. You could say another way, sister chromatids are separated. There's different ways, but that's all that happens, is the whole act of separating the replicated chromosomes. Okay? They've separated. That's all anaphase is. So now the final thing is the last stage, telophase. So in telophase, you are now going to pretty much end the process. So I've got one centriole up here at the north. I've got another centriole here at the south. I've separated my DNA. So now what I want to do is just organize it back into a nucleus. Remember, this is nuclear division. So by the end of this process, I should now have 2 nuclei. And that's what happens. I'm going to draw the top one up here. And I guess you can give yourself a little more room. But notice I now have 2 circles reappearing. This dashed line now means that my nucleus reappears. It disappeared in prophase, it reappears in telophase. So if I look at the contents of my nucleus, early on, I'm going to have those look at how many I have. I have 1, 2, 3, 4. 1, 2, 3, 4. The nuclei, this one chromosome went into one nucleus. This chromosome went to another nucleus. These are replicates of one another. So if one nucleus has this, one nucleus has this, they're going to be identical to one another. They have the same sequence of DNA. This nucleus right here will be genetically identical to this nucleus right here because they are all copies of one another that have been split. So telophase, the nucleus reappears. What also is going to happen is, now that the replication process is over, the chromosomes will uncoil. So really, by the end of telophase, this is kind of early telophase, because you still have chromosomes. By the end of telophase, this is what, you don't have to draw it, but I just want to show you what will happen. These chromosomes will start to uncoil and will turn right back into kind of that sloppy spaghetti, because it turns back into chromatin. It's no longer needed to be in that form of chromosome because the process is over. So now we have two nuclei that are identical to one another within one cell. Alright? Two nuclei, one cell. And now the last thing that is in the cell cycle is the third phase. Cytokinesis. So by cytokinesis, we have a full nucleus back, completely in chromatin form. There's my top centriole. There's my bottom nucleus, completely in chromatin form. There's my bottom centriole. This forms, cytokinesis is the splitting of the cell. In animal cells, they start to pinch together. You see this action of the cell membrane begins to pinch. It's called a cleavage furrow. You can foresee what's going to happen. This cleavage furrow is going to pinch even more and more and more until you would have two distinct cells. These are my daughter cells now, genetically identical to one another. Daughter cell 1, daughter cell 2, half the size of my original parent cell. Okay, that's today's lesson. By tomorrow, I want you to be able to recognize distinct things that happen in each phase. You may see some type of assessment tomorrow that will judge if you can do this. You should have some basic knowledge. I don't need to memorize everything, but if I ask you to say what is one thing that occurs in each phase, you should come tomorrow being able to do that. All right? I'll see you then.